Hello everyone and welcome back to Armstrong's Laboratory, the channel where we break down academic papers so that the information is more widely available to everyone. In this video, we are looking at the paper, The Expression of the Suicide-Associated Gene, SKA2, is decreased in the prefrontal cortex of suicide victims but not of non-suicidal patients. Throughout this paper, we will be looking at SKA2's role in suicide. Nearly 35,000 people are dying by suicide every year in the United States. To study suicide, researchers typically utilize post-mortem brains of suicide victims. Researchers think that DNA methylation plays a large role in suicide. If you watched my last video, you know that DNA methylation decreases gene expression by the addition of a methyl group, which blocks the machinery to make RNA. One study found a specific methylation site on a gene known as spindle kinetochore-associated complex subunit 2, or SKA2. Blood samples from postmortem brains have been shown to have decreased SKA2 gene expression in suicide victims. This may be potentially due to the methylation of SKA2 DNA. Decreased SKA2 gene expression was seen in suicide victims who suffered from depression and were violent suicide completers. Violent suicide completers refers to the violent method, such as a firearm, that was used to complete suicide. Current evidence seems to suggest a dysregulation of the expression of SKA2 in the blood of suicidal individuals, and may be a potential indicator of individuals who are suicidal. While studies have looked into SKA2 gene expression in individuals who commit suicide, researchers cannot conclude if SKA2 downregulation is seen in suicide victims or just patients who suffer from major depressive disorder. These researchers needed to conclude if SKA2 downregulation was specific to suicide. To assess if SKA2 is specific to suicide, the researchers measured both the gene and protein expression of SKA2 in the prefrontal cortex of individuals who suffered from suicide but did not have major depressive disorder, and those who died by suicide and suffered from major depressive disorder. They also included a schizophrenia suicide group and a schizophrenia non-suicide group. They also had another group that suffered from drug abuse disorders and committed suicide, and a control group which did not commit suicide or suffer from a psychiatric disorder. Gene expression refers to the DNA being transcribed into RNA, while protein expression describes RNA being translated into proteins. This is very specific language that all researchers use when talking about gene and protein expression. To determine these forms of expression, these researchers used both qPCR and Western blot. For our purposes, we just need to know that qPCR measures gene expression by amplifying SKA2 DNA, and that Western blots measure SKA2 protein expression by utilizing antibodies. If you want a more detailed explanation of these two techniques, I have included links in the description that go over them in a little bit more detail. The first thing that this paper discusses is the demographics of the individual brains that these researchers utilize. You can find the specifics in the paper, but here's a quick summary of the information available. As you can see, a majority of the brains used in all of the groups were male, with the exception of the major depressive disorder suicide group. The number one killer for groups that did not commit suicide, regardless of psychiatric disorder, was cardiovascular disease, and I don't think that comes as a surprise to anyone. In the suicide major depressive disorder group, these individuals died mostly by drug overdose, although this may be due to the large amount of female brains in this cohort, considering females are more likely to commit suicide by overdosing than men. In the schizophrenia group, the most common method of suicide was falling to their death. In this other suicide group, gunshot wounds were the most popular method for committing suicide. You can find more in-depth information about the individuals and their brains, such as what drugs they were taking, in the paper itself. Now that we know a little bit more about the demographics, let's take a look at the results. First, let's look at the control group. This is a group that did not suffer from a psychiatric disorder and did not commit suicide. We're going to use this group as a reference to observe their altered levels of SKA2 gene expression in our other groups. When comparing all the non-suicide psychiatric disorder groups, we observe a similar level of SKA2 gene expression to the control group. No psychiatric disorder non-suicide group alone displayed significantly less SKA2 gene expression. Now, when we combine all the suicide groups together, we see that SKA2 gene expression is lowered compared to both of these groups. This can even be further broken down by psychiatric disorders. Let's take a look at the suicide schizophrenia group. We see less gene expression of SKA2 in this group. Although statistically significant, it isn't as low as the first bar graph that we saw initially. Now we incorporate the other suicide group, we see even less SKA2 gene expression. Then when we add the suicide major depressive disorder group, we see even less SKA2 gene expression. So while all suicide victims with psychiatric disorders have SKA2 gene expression decreases, major depressive disorder seems to show the biggest decrease. Although it is important to note that while all three groups did show a decrease, there was no statistical significance between the three of them themselves. Now let's take a look at protein expression. Some of you are probably making the assumption that if gene expression is decreased, then protein expression is decreased. Well, you'd be right in this case. The results showed that there was a decrease in SKA2 protein expression in all the suicide groups compared to all the non-suicide groups. It didn't matter what specific psychiatric disorder from the suicide group the researchers were comparing to the control groups, as all three had similar levels of protein expression. So both the gene and protein expression data are showing that there are decreased expression of SKA2 in the prefrontal cortex of suicide victims with psychiatric disorders, but not psychiatric disorders who didn't commit suicide. This data seems to suggest that SKA2 downregulation may be specific to suicide and not psychiatric disorders. So while the researchers here have shown that SKA2 may be suicide specific, no one has yet to show how SKA2 really fits into the bigger picture of the biological mechanism of suicide. So trying to gain a little bit of insight, researchers looked at what we currently know about SKA2. SKA2 belongs to the SKA complex, which consists of SKA1, SKA2, and SKA3. This SKA complex is required for cells to progress from the metaphase stage of mitosis to the anaphase stage of mitosis. These are specific phases of cell division that all of the cells in your body undergo. There are a lot of videos on mitosis if you're looking for a more in-depth explanation of the process. What we just need to know is that without SKA1 or SKA2, cells take nearly 5 to 6 times longer to complete mitosis. Interestingly, though, when SKA2 is overexpressed, the amount of mitosis occurring increases. So SKA2 may be playing a role in mitosis a little bit more than some of those other SKA proteins. SKA3 is thought to be responsible for stabilizing interactions between parts of the chromosomes and the microtubule, which are very important for metaphase. Without SKA3, mitosis even fails to occur at all. It is thought that SKA2 is involved in suicide by its interaction with the glucocorticoid receptor, the receptor that processes stress hormones. This receptor also plays a role in regulating the stress pathway known as the hypothalamic pituitary adrenal axis, or the HPA axis. The glucocorticoid receptor works through this pathway to inhibit the production of intermediate hormones that produce stress hormones through a negative feedback loop. In suicide victims, there is a hyperactivity of the HPA axis, which may be impacted by the glucocorticoid receptor inhibiting that negative feedback loop. Glucocorticoid receptors are downregulated due to early life trauma through DNA methylation, and this may inhibit the negative feedback loop. Studies have shown that cells with stress hormones, SKA2 will interact with the glucocorticoid receptor to move to the nucleus. But with decreased levels of glucocorticoid receptors, SKA2 is unable to move to the nucleus. The interaction between SKA2 and glucocorticoid receptors is also decreasing gene expression. This interaction will interact directly with DNA to begin the expression of genes through the process known as transactivation. With a decrease in SKA2 expression, this could lead to further decreased interactions between SKA2 and the glucocorticoid receptor to decrease transactivation. SKA2 itself is regulated by staurosporine, formylestrin, and trichostatin A. These are three molecules that induce the destruction of cells or inhibit cells from dividing. Each of these three molecules have a specific pathway that honestly warrant their own video because they can get a little complex. To kind of sum up their pathways, we just need to know that they work through protein kinase C, which is responsible for making modifications to proteins. Other research has already shown that there's abnormalities in protein kinase C in suicide victims, so this may or may not be related. So right now we know that SKA2 is being regulated by glucocorticoid receptors, protein kinase C, and the HPA axis hyperactivity. This may be causing decreased cell division and through its interactions with the glucocorticoid receptor or through protein kinase C, or maybe a little bit mix of both. These interactions may be responsible for the volume and structural changes in the prefrontal cortex, which is observed in the suicide victims' brains. Thank you for taking time to watch the video. I hope you enjoyed it and learned something along the way. If you are looking for more content, please subscribe to the channel and leave a like on this video. We will be uploading videos weekly on the topic of suicide and understanding its biology. Please take some time to check out my other video and feel free to leave questions in the comment section. Thank you.